Warmly welcome to this EVOLTREE webinar. Today we have 3 exciting speakers for you, but for those of you who might be joining us for the first time, I will say something about EVOLTREE. So, EVOLTREE is a network that is more than 10 years old, and it's focused on evolution in trees and associated organisms. It provides networking, travel and project grants, conferences and seminars. And if you want to have more information about how to access these grants or conferences and seminars, please visit our website at evoltree.eu. And then before I continue, I think I have to introduce myself. My name is Mariana Westergren. I work at the Slovenian Forestry Institute, and I will be moderating this webinar. And as I said, we have 3 speakers today for you. Dr. Jaime Huerta-Cepas, he will be talking about phylogenomic approaches to analysis of evolution and functional novelty in microbial communities. And he is a group leader at CSIC, working on computational methods and genomic resources to characterize microbial communities from a functional and phylogenetic perspective. His lab is particularly interested in the characterization of novel functional modules associated with specific environmental conditions, for which they combine evolutionary analysis, ecology, massive sequencing data, and high-performance computational resources. And the lab is currently focused on projects in comparative metagenomics, phylogenetic diversity within microbial communities, evolution at the gene family level, and phylogenomic methods and tools. But before I give the word to Jaime, we will have 2 flash talks. First will be by Siki Deng, and he will be talking about PhiloCloud, an online platform for making sense of phylogenomic data, followed by Claudia Sanchez-Lopez, who will be talking about chemoreceptor profiles in plant-associated bacteria. So, Siki, the floor is all yours.
Okay, thank you. Now I will share my screen. So, is it working? Okay, yes. So, hello everyone. I'm glad to be here. My name is Siki. I'm currently a PhD student in Jaime's lab. Today, I would like to introduce our latest project of this year. It's called PhiloCloud. It's a web server for analyzing phylogenomic data. So, as we know that phylogenomic data have grown exponentially over the last decades, and it's very common for the genome-wide project to generate hundreds, even thousands of phylogenetic trees with a multiple sequence alignment. So, which will be maybe also very large in size. So during our research, we encounter the following challenge we hope to solve in the analysis. First, the tree can be very large, and because of the more and more the data we generate. And currently, we lack of comprehensive software where for the easy management, facts explorations, especially for those large annotated phylogenetes. Here, we're talking about like more than 5,000 tips tree. And second is we generate a lot of data, so we have too many trees, and we don't have a like solid platform specific for large size of phylogenomic data storage and management, including their multiple sequence alignment. And the last, it's most of our phylogenomic analysis still rely on custom bioinformatic workflow, which require programming background. So we hope to have more user-friendly software. We can do that. So here is one of the examples from our research. This is a tree more than 7,000 tips in the one of the phylogenetic viewer. As you can see from the right-hand side, when we zoom into the branches, it become collapse and almost unreadable. So based on in order to solve this challenge, we developed PhiloCloud, which is an online platform for interactive analysis, visualization, and manipulation of a phylogenetic tree to allow users to handling large collection of trees, virtually any size, and providing easy access to common operations. And we luckily to already publish this project in NAR web server issue earlier this year. So it's already welcomed everyone to use to test it. So here we have the major scheme of a PhiloCloud. So here, for example, the users have a phylogenomic project where contains the trees and alignment over one single click will save and store into a tree collection in PhiloCloud. Each tree will generate a profile, and all the tree will be automatically indexed for the further query. And about each tree have a tree profiler will be visualized by the ET tree engineer. So in the top right hand side Explorer pattern here. So ET toolkit, it's a Python library. It's a Python library package for tree visualization, which also developed from our lab. So Jorge and Jordi, we are developing on the ET4, the version 4, hopefully to release in the next year, and where we are able to fast explore the large tree and also do all the actions. And PhiloCloud, we use it as a viewer, so users can browse and do also to editing on all the tree and branches and all the data. And apart from this, PhiloCloud provides a handful of a tree annotation and analysis tool, so you can interactively analyze the tree in the browser. So including taxonomy annotation and also, for example, evolutionary event detections. So, and all those actions can be saved back to the tree collection, back to your database, and where you can and also you can share with your private via private link to your colleagues or collaborators, or decide to share in public. So here we have one of our short video to demo how the interface look like, and also the tree annotations. So here is the tree that we saw earlier before and here is with the tree alignment. So you can see just one click, and here attached the alignment will be showing on your right panel and alongside with the annotated tree. So you can see the zoom, and also it's very seamless. It's no delay. And here we show another example is about how to use the tree annotation. So you can see as all the tree tips here, it have the NCBI ID and the gene names. And here we choose the NCBI annotation or GTDB annotation. And then we will only one single click. We can see all the tree annotation will be showing the ranking. It's on the right hand side, and evolutionary events is already in the internal nodes. And here, as you can see, now the properties are already it's annotated as a taxonomy information over all the tree nodes, and including the evolutionary events. This is so this is like a basic demo of the PhiloCloud, and of course, due to the time limits, we cannot. There is some. I'm not able to show more for the PhiloCloud, but here is briefly introduce of what we can do more. It's we host currently we have like the ECNOC 5. It's a taxonomy and a functional annotation database in the PhiloCloud we can open to explore, and also Spongilla data, and from the Musa publish earlier this year. Sorry about the time, I said it wrong. And also a handful of phylogenetic tools, including phylogenetic tree construction workflow, taxonomic tree query, and a tree comparison and visualization. And also there is a more handful tool we are under developing. So so here we thank you, Jaime, to introduce me these projects, and also Jorge and Jordi, who is the engineer behind the tree weaving engine, and also all of my colleagues to provide all the constructive criticism. Also, thank you for my funding, which is from the FFAE and then Chan Zuckerberg Initiative. Thank you very much.
Thank you, Siki. This looks like a really useful tool. I'm sure there will be, I'm sure there will be a lot of questions, but I will ask you, all of you in the audience, to hold the questions now or write them down, and you will have a chance to ask Siki everything at the end of the third talk.
Okay.
So, Siki, please stop sharing your screen. And
no problems.
Yeah, and we will give the word to Claudia. Claudia, please.
Can you see my screen? Sorry.
Yes.
Okay. Then, okay, I will present our last genomic analysis called chemoreceptor profiles in plant-associated bacteria. And I'm also a student in Jaime's lab, but also I'm sharing another lab that is specialized in phytopathogenic bacteria. So first of all, I'm going to introduce you which are the chemoreceptor proteins that are a family of proteins very important for bacteria, as they play a key role in the adaptation to different conditions. They are sensing cues of the environment with the sensing domain that is a extracellular domain that binds to different nutrients or toxins or different conditions, and it has a cytoplasmic domain that is called MCP signal that generates a cellular response that could be, for example, motility to that nutrient or against that toxin, or also biofilm formation, among others. So the study of chemoreceptors have been done in a lot of model organisms, and it is known that they are essential for making different interaction with bacteria, with other hosts, and they are important in complex habitats. The plant habitat is very known, and it is known that they are important in the colonization of the plants. But although the adapting role seems to be very important, and it's quite clear, it haven't been proved the ecological importance in a global scale. So in this work, we wanted to characterize the global landscape of the chemoreceptors, focusing our attention in the plant-bacteria interaction. So for avoiding that, first of all, we worked with the progenome dataset that contains almost 12,000 of species representative genomes and extracted the chemoreceptors sequences. We created a catalog with almost with more than 80,000 of chemoreceptors. But as I told you before, the ecology is settled more because of the sensing domain. So we extracted also the sensing domain from this catalog, designing our own bioinformatic pipeline. And we obtained more than 70,000 of sensing domains. That one of the main advantages were that we were including those that were previously uncharacterized. And with all these sequences, we classified them, clustering them by sequence homology. And with that, we created families of sensing domains that a priori are sharing a common evolutionary origin. So with these 5,000 of families, we wanted to know which of them were more important in the plant associations. And for that, we analyzed the initial data set and extracted the species that were associated with plants. This was a manually reviewed work, and with that, we could identify which were the sensing domains present in the plant-associated lifestyles. One of the main results, and also most interesting, was to know that the majority of the families that were enriched in these plant-associated species were from the unknown types of sensing domains. And also, one of the analyses we did was to know if we could use these sensing domains as potential biomarkers of the plant-associated lifestyles. And for that, we measured the phylogenetic signal, because if the case was that these sensing families were only evolving according to phylogenetic signal, this would be the example, we couldn't say that they are good biomarkers, because the habitat weren't playing a role in the evolution. So we wanted them not to be following a phylogenetic signal. And when we analyzed the plant-associated sensing domain families, we saw that the majority weren't following this phylogenetic signal. So we could say that the evolution of the families was driven, at least partially, for this ecological pressure that could be that they all were in the plant-associated lifestyles. So to conclude, the take-home messages would be that we carried out a phylogenomic analysis of the full repertoire of chemoreceptor, and we have identified sensing domains that are abundant in these plant-associated bacteria. Also, we analyzed the phylogenetic signal, and the lack of this signal could be explaining that the sensing domains are partially driven by ecological pressures. And only to mention, we are now working on apply this pipeline also in metagenomic data, and to see if this conclusion could be maintained in different environments. So thank you all, and thank you to my both labs and for your attention.
Thank you, Claudia. Very interesting work.
Thank you.
Also for this, please hold your questions. So we give the floor now to Jaime for a slightly longer presentation.
Yes, let me share the screen. Can you see my screen now?
Yes.
Okay, cool. Okay, well, let's go. First of all, thank you very much for the invitation to give this talk. I must say that in the beginning, I thought maybe I was not a perfect fit for your community, because yeah, we do work a lot on evolution and trees. It's just a different kind of trees, you know. It's our trees live mostly in our computers, and everything we do is basically computational work. But in any case, Stephen told me that this could be, anyways, very interesting for your community. So what I'm going to tell you today with this presentation is a little bit of a broad perspective on what are the phylogenomic approaches that we are applying at the metagenomic scale to analyze microbial communities, and in particular to investigate what is the evolution and functional novelties that are revolving about these microbial communities. So as I said, we are a bioinformatics group, so most of our work is in the dry lab, in the computer. We do a lot of data analysis, mostly driven towards questions that are ecological questions, or evolutionary questions, or functional prediction questions, and for which we end up developing a lot of novel tools and methods, basically because the questions that we are trying to address are not possible with the current software, so we end up developing a lot of new software and tools, etc. And the little part of wet lab that we do is basically to generate data that maybe is not available in public repositories, and we want to explore certain environments or generate our own samples, and this is the only part that we do in the wet lab. So that we are based at the CBGP Institute, which is a plant biotechnology and genomics institute, although we do not work directly on plants, only indirectly, for instance, with projects like Claudia's project, connecting microbiome with plant-related biology, etc. But most of our work, at least presently, is about the analysis of associated microorganisms with plants and with many other environments. So our focus is metagenomics, but we address metagenomics from an evolutionary point of view, and in particular via comparative genomics method. So I thought it would be interesting to introduce very briefly what we understand by comparative genomics and what are those methods that we are trying to apply at the metagenomics scale. So probably all you know about these concepts, but comparative genomics is basically the field where we are trying to extract evolutionary information from genetic material based on conservation patterns or comparisons between genes and proteins from different organisms. And the canonical example or the classic example of a comparative genomics analysis would be a molecular phylogeny, where we are aligning sequences from different genes from different organisms, and we do a multiple sequence alignment from which we can generate a phylogenetic tree and extract evolutionary information from there. So obviously we can extract information about how the different species are evolving based on these comparisons, but there are many more things that we can do with comparative genomics. We can test for a specific hypothesis regarding selection pressure, so we can identify what branches or what species are under more or less selection. We can even identify the specific sites and residues that are under selection, and we can extend these comparisons to even larger analysis or different analysis, like it could be synteny analysis, or how is the genome architecture conserved across evolution, so genomic context or synteny analysis, etc., for which we obviously also need this kind of comparative analysis at the sequence level. We can do comparative genomics at the protein structure level also to find similarities, and we can use comparative genomics to trace biological events or evolutionary events like gene duplication and speciations, and then track specifically sub and neo functionalization events happening at a very specific point in evolution and affecting specific genes, etc. So this is a general view of what we can do with comparative genomics, and comparative genomics is a field that has been developed for decades now. So there are a lot of softwares and methods and approaches to address all these questions. There is one central concept also associated with comparative genomic analysis that is very important and that we work a lot about, which is the concept of orthology and paralogy, which is basically trying to disentangle two different ways of evolution, or two different evolutionary ways of diversion that sequences, homologous sequences, could follow. So all we have seen in the previous slide would refer in principle to homologous sequences that, bioinformatically speaking, these are sequences that we can detect with sequence similarity searches like BLAST or any alternative software for this. But if we are meant to extract more meaningful information, evolutionary information about those sequences, it is very important that we can distinguish sequences that are evolving after a gene duplication event, that would be called paralogs, or sequences that are diverging after speciation events. This is called orthodox. These are technical terms that are used a lot in evolutionary biology and in molecular phylogeny, and although it is sometimes forgotten, they are behind many of the methods that we are using for functional annotations, for species tree reconstruction, etc., etc. Obviously, the real scenario is not like the one that you are seeing here. Phylogenies and phylogenetic trees are much more complex, and this is leading to very complex scenarios where the orthologies and paralogies are nested in a hierarchical way, and there are co-orthology relationships, co-paralogy, masked paralogy, paralogs, interest between different species, etc., etc. But fortunately, there are, as I said, many methods and even software and implementations to do this in an automated way. We have had many years to develop these methods, and we could say that this is pretty much established. The importance of distinguishing this orthology and paralogy, which is a central concept in many of the things that we do in our lab, is because if you can identify orthologs in a more accurate way, it means that it will affect your downstream analysis somehow. And there are many downstream analyses that depend on this kind of delineation of sequences, of homologous sequences. So the first one, and it has huge implications, is for functional prediction. So the first thing we have to know is that when we are trying to understand or predict what is the putative function of a sequence, it is much better if we base our comparative analysis on orthology assignments than just homology assignments. And this is something very well known and studied. It's not a golden rule that applies always, but this is, at least in the field of orthology, this is something very well established. This functional conservation of orthologs is very well illustrated by some recent work where you can see how very distant orthologous sequences can replace each other. So this is a very nice and illustrative example of how human genes could replace their orthologs in yeast, even though they are separated very far away in evolution. The second highlight for orthology calling, or the second application for orthology-based analysis, is that they are the preferred content to analyze the gene content of different organisms or genomes, let's say, which is a central idea when you are working in prokaryotic or in microbial genomics, let's say, because there is this concept of pangenome, for instance, that depends heavily on the definition of gene content and how you identify gene content. So this idea of identifying a gene in a genome implicitly incorporates the idea of an ortholog. When you are trying to find gene A in genome 1 and genome 2, what you are trying to establish is that these two genes that you call gene A in both genomes are orthologs with each other. And how you infer this orthology and how you use these predictions for pangenome analysis or microbial genomics analysis in general is very important and has some implications. So I'm putting here one example of a very recent work done by Sayoa here at CBGP, where we actually analyze a little bit the implications of one method or another method for orthology calling when it comes to pangenome analysis. And the third obvious implication of orthology is that orthology is what we want to use, ideally, if we want to identify a species and place them in an evolutionary tree, in a phylogeny, right? So when you are establishing the phylogenetic tree of a given set of species, what you want to use for those phylogenetic trees are single copy orthologs, let's say, right? So it is something very important for all these generic applications. All this, as I said, has been developed for many years now, and it is possible to be done at the gene family level in a relatively easy way. It is a bit more challenging when you try to do it at the genomic scale, not even metagenomic. Here I'm talking only genomic. So we have worked a lot on this, on the possibility of doing this kind of analysis, this orthology delineation, functional analysis, etc., at the genomic scale, meaning that we automate the process of building all these phylogenies, identifying the orthologs, and naming the genes. We have done this essentially. We have done this for different species and genomes. This is one of the most recent work that we have addressed, in collaboration with Detlef Arendt at EMBL and Jacob Musser. So what we did here, Anna did this in our lab, was addressing the problem of naming genes in a new genome, the Spongilla genome, where it was particularly difficult to establish orthology relationships between some of the gene families that were very important for the study that we were addressing here, which is the evolution of the nervous system. And it was basically very important to have good names, good orthology calling for some of the genes involved in presynaptic and postsynaptic processes. Obviously, this has nothing to do with plants or trees, etc., but illustrates very well that this methodology, automating the orthology calling and functional prediction and just finding the correct gene in a new genome, is very important and can change a lot the interpretation. In this case, what we found is actually that some of the genes that were meant to be absent in the Spongilla were actually there, just very distant and hard to identify from an orthology perspective. So for this, we automated the reconstruction of thousands of molecular phylogenies. We analyzed them automatically, and we established this orthology relationship. And then from there, we can start analyzing these genes in the context of their correct gene family and do all the comparative genomics analysis that I mentioned before. we can scale even more this kind of analysis from a phylogenomic perspective, and this is what we are also doing in the lab for model species, or for at least for reference species. So in the lab, we maintain and develop this database called EGNOC, for which we have released a new version very recently. Also, this is a work led by Anna and Damian. Damian is in ETH Zurich. And here, basically, we are trying to maintain a database where a huge number of organisms and genomes, in this new release, we have more than 12,000 organisms, are analyzed from this phylogenomic perspective. We are identifying orthologs, we are identifying functional profiles, we are annotating all these genes, and we are building the phylogenies, alignments, etc. for you. So this is in a database, and you can look at. So all this introduction about comparative genomics and orthology, etc., is just to illustrate that this where we are at the moment in terms of scalability and the possibilities that that comparative genomics is giving us at the very large scale. But actually the topic of the talk is how can we scale all huge amount of methods and tools that exist already in comparative genomics and that are very powerful to the analysis of environmental DNA. So this environmental DNA basically links to microbial community. So if we do metagenomics, and I'm referring here to shotgun sequencing of environmental DNA, not metabarcoding. So if we do this, and we, instead of sequencing a single genome, we have a microbial community sequence, how we can apply these comparative genomics methods to extract the most interesting evolutionary aspects out of it. And as you can imagine, if this is not done yet, at least not in a very standard way, is because it's quite difficult. And this is where we are actually now more focused in the lab. So the main problems and challenges for addressing this kind of phylogenomic analysis at the metagenomic scale are both technical and biological, we could say. So technical, because we are sequencing complete microbial communities. So that means that there are many organisms in the same result. This the DNA of these organisms is obviously fragmented, very much fragmented. They are mixed one with each other. There are different abundances, and very importantly, they come from organisms that are not necessarily known organisms. And this is one of the main challenges that we are addressing in this kind of analysis. So most of the sequencing data that we are producing in this metagenomic analysis is from unknown organisms. And that means that the genes that we could assemble out of this data are also unknown. So this plot here, taken from this reference, illustrates very well how the proportion of hypothetical proteins distribute in known phyla. Okay, so these are genomes from that. These are sequences coming from fully sequenced genomes from known phyla in the reference databases in RefSeq or whatever database you can think of. And this is how it looks, the proportion of hypothetical proteins in the case of candidate phyla, which is unknown. So you can see that the proportion grows a lot, and most of the genes in this case are hypothetical proteins. They are hypothetical proteins because of many reasons that we will discuss later, but essentially because there are no representative species in our current reference databases that can be used to transfer knowledge. So we can actually, for many of those sequences, we cannot do comparative genomics because we have nothing to compare with. This problem of unknown species and unknown genes is not neglectable at all. This is pervasive in any metagenomic analysis, and even though we can extract a lot of nice information and useful information from microbiome analysis in the community, we are all aware that we are ignoring a huge amount of data, basically because we don't have the tools and the resources to analyze them properly. So here in this plot, you have a representation of how this metagenomics data look from a functional perspective. So usually there is a large proportion of sequences that we can identify for which we can identify homologs in reference databases, and also we have some functional information from the homolog, so we can use this orthology calling, or at least homology searches to annotate the sequences that we obtain in the metagenomic survey. Then there is a proportion of sequences in metagenomic sequencing that links to hypothetical proteins in the reference databases. These are functionally unknown, but at least we have a link, we have an homolog in our reference databases, meaning that there is at least one fully sequenced genome, let's say, in reference databases to compare with. Then we have another fraction of sequences, which is very large in terms of number of genes and proteins, which is basically representing sequences from uncultured species that cannot match any protein or any gene in reference databases. This means that you blast any of these sequences against NCBI, for instance, and you will get nothing. And the proportion of genes that behave that way is huge. It's huge for almost any kind of microbiome analysis that you perform. So in the best case, like human microbiome that is more studied, etc., you could get like 20% of sequences matching this category. But if you go to soil or to more biodiverse environments, or extreme environments, etc., you can reach percentages like 50 or 60%. That means that 50% of your reads, the DNA that you are sequencing, cannot be tracked from an evolutionary point of view. And what we do with this, basically, we ignore this data in most of our analysis. And then there is a third category, very important. I will not talk today about this, but there is this category of undetected unknowns. These are sequences from organisms that we know that they are there, but they are in low abundance, and we cannot even sequence because the depth of sequencing is not enough, and we cannot see them. They are extremely important for some environments, so they are known to be part of geochemical cycles, and they are biologically and ecologically important, but we cannot even sequence. So we basically know that this is a huge fraction, but we don't know even how big or what to expect here. So today I'm going to talk about this fraction from here. So where we are in the analysis of these unknown sequences, and what are the main challenges and opportunities and problems that we have found analyzing this data. So as I said before, until very recently, this was the two possible fates of genes and proteins that we are producing in microbiome analysis, in the analysis of microbial communities. If the sequences that we can assemble their reads or hopefully more than reads, full-length genes, etc., were matching reference databases like NCBI, ECNO, RefSec, whatever you want, then we have known homologs, and then we can do some comparative genomics with them, analyze them in a proper way. For those that were not having homologs, we were basically ignoring the sequences. So we were excluding them from most of the bioinformatic pipelines and analysis, etc. And as I told you before, this proportion of sequences is huge. Only very recently, and before I explain this, the reason for this is twofold. One, we don't have the proper computational or comparative genomic framework to address these sequences coming from metagenomics. And second, until very recently, we didn't even have tools to perform basic searches like a blast search at this scale. We are talking here about millions and millions of sequences. And this changed recently with some developments like MMSEX or linked class algorithms, which is software that is somehow replacing BLAST, but making it much more affordable. So we can now, this enabled us to do comparative genomics of metagenomic sequences at a very large scale. And thanks to this, there have been some recent efforts to incorporate these unknowns into bioinformatic pipelines, like this recent work from Chiaravani et al., where they are trying to unify the unknown and the known fraction of sequences coming from metagenomics into, let's say, protein clusters or protein families that we can then analyze from a comparative genomics point of view. However, how we analyze those protein families and protein clusters depend on the methods from evolutionary biology or comparative genomics that we can apply at this scale as well. So, and that means that we need to even build the framework to work at this kind of at this depth of analysis. So, and this is something that we are trying to do very intensively in our labs, where we are working at 3 layers of development. First, we are trying to organize the data, both reference data from isolated organisms, but also trying to unify metagenomic data from different ecosystems, etc. So we can do some proper comparative analysis, not only for within habitats, but also cross habitats, and this enabled us to ask questions from an ecological point of view that are more interesting. We are developing tools and methods to address technical things like basic functional annotation based on orthology calling for these metagenomic sequences, or building the phylogenies, analyzing these phylogenies, and identifying novel microbial gene families that could be of relevance from a functional and an evolutionary point of view. So that means that having all this in place, we could, in principle, organize our metagenomic data in different categories depending on whether the sequences have close homologs, and then we can infer functions from reference databases. They are distant homologs. We can at least detect and then play a little bit with phylogenetic reconstruction, etc., to characterize them better. And we organize also the sequences that have no homology at all. So we organize them in protein families, and we, even though we don't know what they are doing, we can do a lot of analysis blindly without knowing what are those gene families. So just to give you a sense of how difficult it is just migrating all these methods to the metagenomics scale, I could mention here just the basic step of identifying what is known and what is unknown was quite challenging until very recently, because these sequence similarity searches were very expensive, computationally speaking, and not to mention orthology delineation for proper phylogenetic annotation. For proper functional annotation, sorry. So this is one of our first efforts in this direction, EGNOG Mapper, which was updated recently by Carlos, also from our lab, where we are basically enabling functional annotation at a massive scale. So thanks to this tool, we are now able to process hundreds of millions of sequences in a timely manner, and then not only annotate the known fraction, but also separate the unknown, and then we identify what are the unknown sequences. The second major effort that we have been doing in this direction is somehow merging all the metagenomic information, or all the information from microbial communities that are publicly available in repositories from different surveys and studies, etc., in a single catalog that is using standardized methods, and that also allow us to compare between habitats in a better way. So this was a huge effort, mostly driven by Luis Pedro Coelho, still when I was working with him at TMBL, but it was just finished this year. So here we try to combine the metagenomic data from more than 30,000 samples covering 14 different habitats or biomes, which led, just to give you an idea of the extent of the analysis, 2 billion open reading frames, so 2 billion potential genes. This is huge. It was very difficult to handle and to even start working with it. So from these 2 billion open reading frames, we managed to compile a set of 300 million, there is an M missing here, 300 million species level genes. These are genes that we could link to, in principle, specific different species, and this was done with an operational definition of prokaryotic species, which is usually 95% nucleotide identity threshold, or 97 in some other cases, but this is an operational definition. And from there, we also applied these methods that I told you before that allowed us to cluster the species level unigenes into protein clusters that could resemble in a better way what we call a protein family from a comparative point of view. And that this key step enabled us to ask evolutionary questions in a better way than just using this species specific genes. So the first so we started playing with these protein families and the catalog in general to ask these evolutionary questions and also ecological questions. So the first finding that struck us was that the majority of these species level genes, these unigenes, were habitat specific. So they were not shared between habitats. This could be more or less expected because genes, as I said, were clustered at 95% identity, sequence identity. But the same pattern, we also found them for protein families. For protein families that were clustered, in this case, at a very much more relaxed identity threshold. So it was obviously more sharing of gene families between habitats than in the case of genes, but still more than 60% of the gene families were habitat specifics, and this is telling us indirectly some indirect story about the potential functional or ecological role of these families. It could be that many of these families that are habitat specific are maybe conferring some kind of adaptation or special adaptation to those habitats for the species that carry the gene family. However, when we applied more elaborate methods for selection testing, and this is something that we were able to do because we in this case we had the protein family clusters and the alignments and the phylogenetic trees for all these families. We were surprised to find that most of the genes were not under positive selection. So that means, and here it is important to remark that I'm referring to positive selection, not negative selection. So in this case, what we were trying to measure is that if the variations that we were observing at the gene level, so the mutations that were happening in the different genes were actually under positive selection or not. In other words, were those variations actually contributing to the fitness of the organisms, or they could be considered neutral. What we observed is that for the full majority, and especially for the very rare genes, the ones that were habitat specific, were actually we couldn't find this positive selection signal, which is somehow contradicting or difficult to reconcile with the previous reflection that I made before, that maybe some of these gene families and these genes are conferring functional novelty that is somehow an adaptive character for species in specific habitats. So what we actually realized, and the way that we reconcile these two ideas was thanks to this analysis, where we also analyze how these different genes were actually clustered into gene families. And here it was surprising to see that most of the genes, most of the genetic variants at the species level were actually clustered at a very tiny fraction of gene families. So all these genes that were mostly neutral variations, they all belong to the same gene family. So what we are seeing in this huge metagenomic catalog is a lot of variation that is neutral variation within a very small set of families that are also known families. So they are not, in general terms, they are not unknown families. And the actual novelty comes in this long tail in the distribution of protein families, where we have very small gene families or protein families with few members and a lot of singletons, where if we analyze from a functional point of view, most of them are novel according to the definition of novelty that I explained before. So no homologs in reference databases. So we believe that these are actually the ones that could be conferring some actual functional novelty and adaptation from an ecological point of view as well. But of course, just with this analysis, we couldn't make sure that this is the case. So we decided to continue this study, more specifically analyzing these novel gene families that we were obtaining. So the question that we were trying to solve after this work was, are novel microbial gene families relevant from a functional and evolutionary perspective? We don't actually have the data to say so, basically because all these problems that I mentioned before, that we have no ways of analyzing this unknown data. So we started analyzing these unknown sequences from a comparative point of view, from an evolutionary perspective, if you want, and being very strict to see if they, even though we don't know what they are doing, if they pass all the filters that qualify these gene families as functionally relevant or evolutionarily relevant gene families. So in this case, what we did was extending our catalog. The GMGC catalog that I mentioned before was extended, and instead of using just a gene catalog, we used only genes that could be assembled in a metagenome assembled genome, which is in metagenomics. This is an approximation to a fully sequenced genome. It's not the same quality, obviously, but at least it gives us an idea of what is a genome and a species in our catalog. We use 150,000 metagenome assembled genomes, which is roughly including 400 million genes from different catalogs, different habitats, etc. And then we started analyzing with all the powerful tools that I mentioned before in my introduction of the seminar in the comparative genomics field. So first of all, we tried to improve the open reading frame prediction, correcting for genetic code. That was a technical problem that that was affecting in some cases. Most importantly, we ran deep homology clustering with strict filters for coverage and also for species content. So in this case, because we have metagenome assembled genomes, we had a taxonomy classification, if you want, for all these genomes. So we could identify classes that were involving 3 or more taxa, and that means that they are sequences that couldn't be considered, shouldn't be considered orphan genes because they belong to several species, and they are clustered together, so they are homologous with each other. So this is our first filter to know that these are not spurious sequences. They are in principle biologically important. Then we wanted to decide to study only those sequences that have absolutely nothing to do with the reference databases. So we map against EGNOG with EGNOG Mapper, as I explained before, but also to RefSeq and even PFAM. So we were excluding all the clusters and sequences that were matching even a domain that has been described in databases. From there, we built the phylogenetic trees and the alignments for all these gene families, and we only selected the clusters, the gene protein families that contain something that could resemble a novel protein domain, 20 amino acids that, or more that were contiguous and conserved in the alignment. We removed the spurious protein sequences, and together, all these two things also allowed us to define these protein families as orthologous groups. And this is something that we couldn't do before because we didn't even have the information about the species of these sequences. After all this, we were, you can imagine, we were discarding most of the unknown sequences that we were producing. And we applied one more filter that was very strict, but we also wanted to have in place, which is a selection analysis filter. We require these gene families to be under purifying selection. So we selected only the families with DNDS lower than 0.5, and also with some evidence of expressible or being expressible. So after all these strict filters in the known space, which involves, I think it was 30 million protein families, we were able to detect more than 400,000 unknown protein families. This is if you are not in the field, maybe you are not. You cannot calibrate these numbers, but to give you an idea, in EGNOG, all the orthologous groups that are inferred and representing 12,000 reference species looks like this in a distribution of protein families and the gene length. When we include the 40,000 novel gene families that we have detected here, we are doubling the number of gene families. And put in context, this means that all this huge amount of data that passed the strict filters are basically out of our analysis. We are excluding this from most of our analysis, and obviously they must be very important. So regarding how this novelty, potential functional novelty is distributed in the tree of life, we were also able to detect to observe that it was distributed across the whole prokaryotic phylogeny. But it was striking to see that there were some peaks. These peaks represent genomes for which these novel gene families were accumulating. So this is the number of novel gene families in our catalog that co-occur in the same genome of these orders. In this phylogeny, each leaf is a different order. So we believe that this is these high peaks here could be like potentially novelty hubs for which we have to basically analyze a bit in greater detail. Also important, I'm validating the importance of these novel gene families that we detected. We have some ecological analysis. Remember that I told you that most of the genes that we were detecting in the GMGC catalog in this metagenomic analysis were considered habitat specific or rare genes or rare gene families. Well, this pattern is actually inverted for these 400,000 families. In the case of these specifically selected gene families, many of them were actually multi-habitat. Well, obviously they were multi-sample, but they were also crossing habitats, which is very interesting and also reassuring about the potential importance and significance of these novel gene families. When you see that different genes and gene families are crossing habitats, you obviously think on potential confounders, like maybe these families are associated to mobile elements or transposons, etc. We wanted to check this as well, and yes, there is a correlation between the number of habitats and the number of unknown protein families that we are detecting that cross this number of families, both for mobile elements related with plasmids and viral sequences. But still, even in the worst case, the number of mobile elements that are associated to this mobility is just 20%. So that means that most of the families that we have detected here are not necessarily associated to mobility. They are a constituent part of the organism that carries them. So they must be very important from a functional and also from an evolutionary perspective. We know that they are evolutionary important because we required that DNDS was very low, under 0.5, but we wanted to know if there were particular examples pointing to very evolutionary important gene families. And there is a very easy way to do this in comparative genomics, which is by identifying synapomorphic traits. So if we analyze the gene families from this perspective, what we are trying to detect is gene families that are present in all the genomes and all the species in a given lineage, but not in the other lineages outside this clade, right? So if you try to identify this kind of synapomorphism at ranks like species or genus or family, if you want, it is relatively easy to find this kind of synapomorphic traits, but it is very, very difficult to find synapomorphic traits at high taxonomic ranks like phyla, classes, or orders. And this happens also with known gene families. If you try to identify these synapomorphic traits for known gene families, it is very difficult to find them. There are very few. And in this set, we were able to identify almost 1,000 novel orthologous group or protein families that were behaving that way. And actually, those ones were under even more purifying selection than the rest of the families in the set. So the last part that we tried to do here was try to predict the putative function of these unknown OGs. And as you know, for functional prediction, the basic default method for in bioinformatics is biomology. So we need reference data, reference sequences, and we didn't have these reference sequences in this case. So we had to do it with indirect analysis. The most probably well-known method for prokaryotic species to infer function from an indirect way is by conserved neighborhood. So we know that the genes in the bacterial and archaea genomes are very packed, and they tend to organize from a functional perspective. So, and this is known for decades now. It was just not possible to do it at the metagenomic scale because we didn't have the tools to do this. So this is something that we also try to do in the lab de novo. So that was the work of Jorge and Joaquin, where we developed ad hoc tool for not only analyzing and scoring synteny for these metagenomic sequences in their corresponding metagenomes, but also to visualize the result, inspect it, etc. And based on this, this is just an example where you can see this would be one of these unknown protein families surrounded by known genes. So if you know the known genes around and they pass a given score, you can infer that this unknown gene is probably related in the same pathway as the neighbors. So we developed novel tools for this, and we analyzed our data, and we were able to detect or to predict the function of some of these novel gene families. Some of them were actually inserted in operants that are considered central metabolism or even involved in xenobiotic degradation, etc. So they are likely involved not only in accessory functions, but also involved in some kind of core functionality in the cell. Just to finish, this is something that Siki already mentioned in his talk. One of the main challenges for doing this is migrating the tools in comparative genomics to the metagenomics scale, and this means visualizing data and analyzing data at a very large scale. We do a lot of phylogeny and phylogenomics, so we develop also a lot of tools in this direction. PhiloCloud and ET Toolkit are the two main tools that we develop here. And the final goal of all this is obviously, once we have this framework developed and these novel families delineated, incorporated into the analysis of different specific questions that we also have in the lab. So we are pretty much agnostic about the environment that we are analyzing in the lab. We work in host-associated, implant-associated microbiome, like in the case of chemotaxis that Claudia was mentioning before, but we also apply, and we think it will have a direct impact, the incorporation of these novel gene families into human microbiome, obviously in extreme environments where the novelty, the functional novelty is even higher, and also for understanding the ecology of ocean and soil microbiome at a larger scale. So as a summary, if I had to take a few take-home messages from these things that I have presented so far, I would say that environmental DNA contains huge amounts of unknown sequences, and most of them are currently neglected. There are initiatives to start using them, but still they are very scarce. Most of these environmental genes represent maybe rare neutral variants within a small fraction of functionally known families, but there is a long tail of new families that are also neglected and that we have demonstrated that they have a large significance from an evolutionary and functional point of view. So actually this 400 novel orthologous group that we have inferred are phylogenetically broad and potentially functionally important, ecologically broad or inverting the general patterns, and even some specific cases related with synapomorphic traits that could be involved in the very in the very much definition of the specific lineages, unknown lineages. We use genomic context also to predict function, and this could be applied from now on in a more automated way. But there are many evolutionary analysis and tools that need to be migrated to the metagenomic scale in such a way that we can start comparing and analyzing microbial communities in a more comprehensive way. So with this, I will finish. So thanks everyone. Thanks for the invitation. Thanks for listening, and especially thanks to all my people in the group that actually did most of this work, and collaborators and funders. And if we have time for questions and you have any, I'm happy to take it.
Thank you, Jaime. I mean, my head is still spinning from all the numbers you have shown. It's like mind-blowing, and I never had the idea. So the iceberg there picture is really appropriate.
Yes.
And with that, I'm opening the floor for questions. I'm sure there are some You can either raise your hand. Jaime, if you maybe stop sharing your screen,
Yes.
so we can see everybody. So you can raise your hand. You can ask the questions in the chat and I will read them out. And please turn on your cameras because it's much nicer to have a discussion when we can see the faces. There is a question from Iris. Please, Iris, show yourself and ask the question.
Sorry. Hello, everyone.
Hi.
Hello.
Thank you for the presentation. Very good, very, very, very good. And clarified a lot of things for me. I was wondering here, because I work from Mozambique, I have samples that are very neglected in terms of databases. So the major problem that I'm facing now is regarding the primer design and the specific primers that are the best to find the species specifically in arbuscular mycorrhizal fungi, which is my area of work, my field of work. What can you suggest in terms of this part of the whole story behind the bioinformatics and all the analysis that you taught about now? Specifically, when we are talking about species that are very, very underknown. Thank you so much, Jaime. Thank you, everyone.
You're welcome. I'm happy that it was useful for you. So everything I presented here was huge numbers and analysis, but mostly for prokaryotic species. So this was prokaryotic metagenomics. You mentioned that your problem is about fungi, which complicates even more the story because there is little methodology and reference resources, etc. But the problems are probably the same. If you are trying to, you mentioned that you are using, you are intended to use primers to detect those species. This is not exactly the same as what I was mentioning in my presentation, which is scanning functional diversity, etc. If you are using primers, I guess you just want to detect the species that is there. And I guess ITS, which is a typical barcode that is used to identify fungi, is not working very well. So my suggestion here would be that you obviously use your own primers for genes that are very specific for the fungi that is of your interest. I don't know which one is it. I don't know if you have a reference genome already or something that you could identify. If not, it would be a problem. I mean, this is even a problem for prokaryotes. I mean, not even metagenomics, talking about metabarcoding and 16S, it is well known that there is a bias. For instance, archaeal species are not well captured with the standard 16S primers, and you are missing a lot of biodiversity. Just if you use the standard primers, if you modify your primers towards what you are trying to find, everything will work a bit better. So I don't know how possible it is for you to do this type of primer design, targeting your specific species and taxa. If it is possible, this is what I would do. If not, maybe you can always try like a shotgun metagenomics and then analyze everything from scratch. And in any case, if this species of your interest is not in reference databases, then you will need, or I would suggest, to use the novel methods for species delineation or functional delineation or whatever you want to do, because then you are, if the species is very new, you are going to miss a lot of novelty and information from the reference databases. This is what we are trying to solve, not yet solved, incorporating all this novelty into reference databases. But so far, I would say if you are working on something very novel and it's not in reference databases, go back to the basis, go back to the tools and methodology that we use to build the resources, and build your own resource as a reference to analyze this.
Thank you, Jaime. Also, you can post questions for Sigi and Claudia if you want. And in the meantime, and this is really not my topic, so maybe my question is weird, but you said that you measured fitness when you were quantifying positive selection. I was wondering how you were measuring fitness.
Not really fitness. We were just measuring positive selection, which is something that you can associate with fitness, but we don't know anything about fitness.
Okay.
It would be super difficult to identify. But positive selection with the classic models and statistics that you can use for this, it would in principle identify sequences that are being fixed in the population, and the reason for being fixed is perhaps because they are improving the adaptation to specific niches or stuff like this. So that was the idea. But yeah, obviously, it's not a direct fitness calculation.
Okay.
This is very different.
Okay. Okay. Thank you for the clarification. Please,
No
Santi.
problem.
Well, yeah, just continuing with the positive selection and the questions for Mariana. So basically, you are looking for selective sweeps, right? So that means like this is more measures events of selection, and it doesn't account for polygenic adaptation, or is the classical.
Yes, this is what I try to also express over all the presentation, that what we are trying to do is like the basic analysis in the comparative genomics or evolutionary analysis or evolutionary and selection analysis that we can migrate at the moment at the metagenomic scale. But we are far from applying like more advanced methods and analysis at this scale. You could do it for a specific gene families maybe, and then fine. But at a larger scale, this is where we are, and it was already very intensive computationally speaking.
Okay, thank you. I was asking because when we do this for forestry, very often we also find we don't have these signatures of major sweats affecting large regions of a full gene, but we are starting to build more evidence that actually what we are finding is just small changes, adjustments in allele frequencies that are very difficult to detect. So I was wondering if you were doing also some work in that direction.
No, not yet. So the most we can do with by incorporating this novelty at the moment is try to improve the standard metagenomic analysis, like calculating alpha biodiversity or beta biodiversity in microbial communities and identifying patterns, ecological patterns. This is obviously affected by what are the features that you are analyzing there. So if a functional profile is generated taking into account all the novelty, then maybe the differences that you are seeing are more obvious, or you detect differences that otherwise you wouldn't detect with the known gene families and known functions.
Okay, thank you.
Okay, we have a question from Yvette. Yvette, please turn on your camera and ask the question. Oh, we cannot hear you. Please unmute.
Sorry. Hello, my name is Yvette from Mozambique too, like Iris. I have developed a study in woodland here in Mozambique, and we compare the rhizobacteria from different soils in different fire frequencies. But curiously, we didn't note any difference between the three different soil types in the different fire frequencies. And we also didn't identify the species bacteria of these soils. There is some possibility of there are unknown species that could cause these results. No difference between the three different soil samples, and if yes, it's possible that may I continue the study, and what you can suggest for me from your study? How could I improve my study to confirm that results?
So you, I don't know if I understood completely, but what you suspect is that the differences are caused by an unknown species that you cannot even identify.
Yes, because we cannot identify the species, bacteria species. We just stop in the general, and it was the same. We have the same results from the 3 different cell samples and the 2 different file regimes. It was, yes, a little bit strange, but yes.
Yeah, so I mean, it is clearly, if you try to identify the species, and the most you can get is genus identification or family identification, that this is obviously a new organism. I don't know to what extent this novel organism is actually interesting or associated to the conditions that you might be studying. This is up to you to test. But yes, you could perfectly have an unknown species that is responsible for whatever changes that you are trying to track, etc. So something that you can do is try to explore a bit more I don't know if this species you detect in high abundance or low abundance. This would also affect, but I will try to explore a little bit more the surroundings of this species in terms of the phylogenetic surroundings of this species, or there are other close relatives, also in microbiome studies, etc. If you didn't do already this classification of the species, you should do it taking into account all the previous metagenomic analysis. For this, you can use dedicated databases like GTDB or stuff like that, where you are incorporating not only cultured known species, but also all the microbial species that have been detected from metagenomes, but for which they were able to assemble them a MAG, a metagenome assembled genome, and then this would help you to identify the species a bit better if the same species had been identified in other habitats. And that's it, and maybe sequence deeper if you can, to see if you can characterize this species a bit better, this organism.
Thank you very much.
No problem.
Please, Christian.
Yes, thank you for the nice talk. I'm very interested in this function prediction that you showed at the very end. I think we have a similar situation in forest trees. For example, in conifers, there are studies that found 100,000 genes in the genome, but actually we don't know what they are, except for the 25,000 that are present in the Arabidopsis thaliana database, basically. So how did other actually studies showing how these gene predictions, how successful that they are? Are there like validation study that it can show that what you predict is actually also in an experimental setup or whatever? Is it true, or is it just pure prediction?
Well, this is bioinformatics, so everything is a prediction. You have to just understand what are the assumptions and the thresholds that you are using and the methods in the prediction, and then you decide if you believe it or not. So for functional prediction, as I mentioned, there are many different approaches. The one that I mentioned more in detail, the ECNOC mapper and orthology-based functional prediction requires that you have a good orthology assignment based on, in principle, phylogenies, for instance, or clustering methods that are also working well, and that the functional information that you are transferring comes from an ortholog that has experimentally validated functionality. Okay, and this can be done. For instance, in ECNOC mapper, when you use ECNOC mapper in an automated way, you can say, I want to annotate my sequences using only orthologs that have experimental evidence for the function that we are transferring. Obviously, you are going to annotate fewer genes because you will find fewer orthologs with this meeting this criteria, but you are more sure about the prediction. But still a prediction, so you never had the experimental validation of this. So if you can afford using multiple methods, because you are working only on a handful of gene families, 100 gene families, and you can afford looking at them manually, then I would combine methods. So yeah, do ECNOC mapper or check ECNOC or other resources for orthology, but then also try to use indirect methods for functional annotation that are not necessarily associated with orthology. Synteny, although synteny is not the same in eukaryotes, in prokaryotes is working very well, but in eukaryotes I don't know, and in plants and trees I don't know how much the conservation of the genome is maintained. You can check protein structure similarities to find homology at the structural level, and then combine all this information to have the best of your predictions. So this is what we were trying to do also for all these novel gene families, because basically we couldn't have an orthology mapping strategy to annotate them. So then we had to use indirect methods. Genomic neighborhood, synteny, but also you can use phylogenetic profiles, like knowing where these genes are present across the phylogeny. Just the phylogenetic profile of present absence of these genes is informative about the potential function, the location in the genome, the structure. So there are many alternative methods in comparative genomics to predict function. I would combine them all if you can afford, because it's always a prediction.
Thank you very much.
Are there any more questions for either Jaime, Claudia, or Siki? Okay. There is a question in the chat from Yvette again. What kind of samples are used for detection of chemoreceptor?
In my case, what I worked was with reference data. They were from progenomes, and they are from culture data that they extracted, also NCPI, is only reference data. That is the catalog we extracted all the chemoreceptors from. And those data has some annotation, and from those annotation is where we were able to manually review which were on plants or we weren't. The manually review data that we took for the plant-associated bacteria database that we created.
And if I can add some insights on this, what you mentioned in the last part of your talk, that we are extending for metagenomic analysis. In this case, yes, we are using samples on different environments, but mostly phylosphere from different plants and lithosphere. So we are interested in the chemoreceptor profile in these two specific environments.
Okay. Thank you. And Yvette, for you who lost parts of the presentation, there will be a recording available on the internet, and you will be able to look again at all the presentations. So there will be information there for you. And now I don't think I will ask anymore if there is more questions, because we are reaching 6 o'clock almost. I mean, it's 5 more minutes. I would like to take this time to thank all 3 presenters. It was really interesting, and I learned really a lot. I had no idea about the numbers, honestly. And I'm sure the tools that you have presented will come in useful for many of us listening here today. So thank you. Thank you a lot for taking the time and presenting your work.
Sure, not at all. Thanks to you, Mariana.
Yeah,
Cheers.
And the other thing I would like to take the time to do now is to invite you all for the next seminar. I think Christian has a slide prepared, right, Christian?
Not for the next seminar.
Okay.
But it will, yeah.
But it will be again on Wednesday, and again next Wednesday. So same time. And mail will come again for those of you who registered 1 day before the seminar. And the last thing, I would also like to warmly welcome you to the 2nd biannual EVOLTREE conference, which will be held in September next year in Romania. So please stay tuned on the webpage, and I guess the call for abstracts and things like that will be coming out in due time. So with that, if you have any more questions about EVOLTREE, you are free to contact either Christian or me or even Santi, who is leading the network. So this is an invitation. And I guess that is it. So thank you all. And have a nice evening and a nice December, which starts tomorrow.
Okay, you too. Bye-bye. Thanks.
Thank you. Bye-bye.
Thank you very much.
Bye.
Thank you. Bye.
Bye.
Bye.